You're listening to Mammal Watching with Charles Foley and John Hall. You can find other episodes at mammalwatching.com/podcast.
Welcome to the Mammal Watching podcast with me, John Hall in New York.
And me, Charles Foley in Minneapolis.
How are you, Charles?
I am very well, John. Thank you. I got out last weekend to an area in northern Minnesota called the Sax-Zim Bog. And it's only about 3 hours north of Minneapolis, which is where I live. But this place is famous among birders as a place to go and see boreal bird species, and in particular, owls from the boreal forest, including sort of the mega bird, which is the great gray owl, and also other ones like northern hawk owls. And these species are apparently really difficult to see in the summer as they disperse widely in the northern forests. But in the winter, they will congregate in areas of tamarack forest at the edges of their range, the southern edge of their range. And hopefully, or in these areas, they tend to be a little bit more visible. And the Sax-Zim Bog is one of those areas, and apparently birders come from all over the world to try to see the owls and the other bird species there. But more importantly, of course, the place also has some interesting mammals, including the American martin, short-tailed weasels, and wolves. Now, I'd seen the martin there before, and in fact, there's one individual, or at least I'm assuming it's one individual that regularly visits bird feeders looking for peanut butter. So it's a really good place to see this species. And I suspect that if you gave yourself maybe 2 days, you'd stand a very good chance of seeing one. Anyway, I went up there with my daughter Sierra and her boyfriend, as well as another good friend, James Schiffer, who also lives in Minneapolis. And we only went for one night, and there was a major snowstorm while we were there, which somewhat hampered our movements. But once again, I was really struck at how great thermal images are for finding mammals. So what we did was we drove around the place at night and also during the day, and my daughter Sierra would use a thermal imager out of the open window, and she picked up about 8 or 9 snowshoe hares with this device. And now, these animals are in their white winter coats, and they are really difficult to see in the snow and the vegetation. And sometimes we would have one in the thermal, we would put the torch right on it at close range, and we still could not see it. So again, it just shows how useful thermal images are for mammal watching, and particularly when you're looking for species that use camouflage. And I know that you and we both use pulsar models, and I know quite a few other mammal watchers have those as well, but those can get pretty expensive. They're around $1,500 to $3,000. But I was communicating with Todd Pusser the other day, and he told me that he had bought a cheaper model. It's called the Taipan TM10-256, and it's a monocular. He bought it about a year ago, and it had cost him about $600 back then, but they are now available on Amazon for about $450, at least in the US. And Todd said that the range is less than that of a pulsar, which is kind of what you'd expect for something that's a bit cheaper, but it works really well for up to about 100 meters. And after that, you can still see things, but the definition drops off. And frankly, I would say that the majority of the mammal species that I see are within about 100 meters, certainly the small stuff. And so this might be a good model for people to get who are on something of a budget. And I think we'll put the link to this device in the podcast notes, and we'll see if we can also convince Todd to submit a note about the model directly onto your website. But anyway,
yeah, sounds good.
Yeah, and we didn't actually see any carnivores on the trip, but it was fun to get out. And we did see a northern hawk owl, which is not quite in the honorary mammal category, but getting there. And anyway, still nice to see.
Sounds like a great trip. Yeah, funnily enough, I've just been talking with the manufacturers of a thermal device, I suppose you call it, that actually attaches to your cell phone, a lens, and it's made in China. They're going to send me one to have a look at, but it's a few $100, and the videos they had accompanying it make it look almost as good as a pulsar. You just clip it onto your iPhone. So for convenience sake, at least, that would be amazing if it works. I'm looking forward to testing that out as well. So it's changing very, very quickly.
Yeah. And again, I suspect with these, I think the issue with the pulsars is that you can see things very far away, 1,500 meters. But, as I said, many of the animals that you see are, particularly at night, walking around at night, are closer than that few hundred meters. And in which case, some of these cheaper devices might work perfectly well.
Exactly. Yeah, I don't think I've ever seen anything I was interested in that was more than 100 meters away. Everything's always close you're looking for. In forest, at least. Fascinating. Cool.
Yeah. Yes. And how about you, John? I know that you are off to Africa again soon.
I am going to Chad in a few days. I am so excited I might just burst, I think. I don't really know what to expect, but Martin Royal is out there with a group doing a carbon copy of the trip we'll be doing. And he's been sending me a lot of messages filled with all sorts of species. It's far exceeding his expectations. I think he's seen about 70 species already in 2 weeks. He just says it's fantastic. He's been a lot of places and he's blown away. So I just hope the luck lasts and the animals stay around for when we get there. So I'm very excited, and I'll be gone for 3 weeks. So it's a big trip for me.
Yeah, absolutely fantastic. I hope you get the striped polecat out there. That'll be a corker.
So do I. Martin saw 3 last night, so I'm optimistic.
Oh, wow. Okay. We might have to drag Martin back onto the podcast to talk about Chad as well.
I think so. Yeah, we can have a Chad reunion, and make you very jealous.
Excellent. Well, have a fantastic trip, and look forward to hearing more about it when you get back.
Thank you. I'll see you on the other side.
So our guest today is Dr. Roland Kays, who is a research professor at North Carolina State University and the director of the Biodiversity Lab at the North Carolina Museum of Natural Sciences. He is also a research associate at the Smithsonian Tropical Research Institute in Panama, where he has been conducting field work since 1994. Previous to that, he was curator of mammals at the New York State Museum for 11 years, where he worked on canid evolution, including documenting hybridization and rapid evolution of the eastern coyote. Roland was co-discoverer of the olinguito, a new species of procyon from Ecuador, and he co-founded the largest global databases for both animal tracking and camera trapping. He has also pioneered the use of citizen science for monitoring biodiversity through the first US National Mammal Survey called Snapshot USA. He has active field projects in Peru, Panama, Yukon, and Zambia, and is a regular contributor to TV shows and has written 2 popular books, including one that many of our listeners will have on their shelves called The Mammals of North America. And Roland also runs his own podcast, which we shall be hearing about later. Roland, welcome to the podcast.
Great. Well, thanks for having me. It's an honor.
Welcome, Roland. It's very exciting to see you in person.
So if I can just start off, Roland, asking you how you actually became interested in nature and what your career path was into your current job.
Yeah, sure. So when I was a kid, I ran around the woods in Michigan a lot. Was in Boy Scouts, was building forts, going fishing, catching frogs, all those kinds of things. But at the same time, I was really interested in science in school and biology. And for a long time, those were 2 kind of separate things to me. And I went to school at Cornell University and took all the biology classes freshman year, and then got a job working in a lab in a med school back home over the summer, which was a great opportunity, but I kind of realized I didn't really want to work in a lab the rest of my life. And as much as I like some of the questions and ideas, I didn't want to be sort of I started sort of looking for other ways that you could do biology. And that next semester at Cornell, I took mammalogy class, and I feel like that kind of launched me down the pathway. And I kind of wonder, usually the different ology classes are offered kind of one semester, not the other. I feel like if it had been herpetology or ornithology, whichever sort of ology was offered my sophomore year might have set the course. And so, I had great opportunities to do some small mammal trapping and learn about mammals, and actually got a chance to travel as an undergrad down to Panama with a class from Cornell, which was great. So I had a fair amount of research experience and went right into a PhD program at the University of Tennessee, studying kinkajous. And then I, from there, did a postdoc with Bruce Patterson at the Field Museum, working on African lions. And then got the job at the New York State Museum, where I was for a little over 10 years, and then moved down here about 10 or 11 years ago.
What a career. Now, as Charles said, you've done a lot of work on the South American Prasionids, and that's the family that includes the raccoons and the ringtails. Could you tell us more about this work, and also the differences between the kinkajou, the olingo, and the olingito, which you helped discover? They're all quite similar looking in my experience.
Yeah, sure. So, when I was in college and starting to grad school, I was really fascinated with the sort of tropical canopy as like a new frontier, and as an unexplored, relatively unexplored area. So I ended up focusing on kinkajous and olingos to some extent, but it ended up being mostly kinkajous, just as a species that had never really been studied. There hadn't been hardly any field work on it. It was all some captivity stuff, and a few sort of anecdotal observations. And so when I first went to Panama with an undergrad class, we went out with another mammologist named Greg Adler, and we went spotlighting, and we saw kinkajous all over the place. It was great. And so I was like, oh, you can actually just go out and find them. Like, they're not that uncommon. They didn't really run away from you. They weren't really afraid. And so that kind of seeded the idea of studying them in more detail. And so I ended up doing my PhD on kinkajous, and trapped and radio collared them, followed them around, kind of like you would do a primate study, like habituating primates and following them around and looking at them, except it was at night. And so it was a lot harder to see what was actually going on up in the treetops, but they were sort of, most of them, instantly habituated. They just didn't care that I followed them. And I tried not to use my light too much, and blind them or anything. But they didn't really seem to bother me. They weren't bothered by me following them around. So I was able to collect, observations on their behavior, collected a whole bunch of scats, recorded the trees they fed in, how much they moved, all those kinds of things. And we did some olingo work there too. So we caught a few olingos. They were not as common, and they were a lot harder to follow. And I never could really figure out what they were doing in the trees. I don't know if they were shyer or just further up, but I didn't get nearly as many observations. So I ended up kind of focusing mostly on the kinkajous, which had really interesting social behavior. So it turns out the males are relatively social and hang out with each other. But it's kind of a flexible system where they come together, they split up. Sometimes they sleep together. One time I saw like 5 or 6 kinkajous come swarming out of this hole in a tree right at dusk, which was really cool. And the males also play with the young. So one of my favorite observations was watching the kinkajous had prehensile tails, and so there was 2 kinkajous. It was a male and a young male hanging upside down by their prehensile tails, like boxing at each other, just kind of playing, but hitting at each other. That was really one of my favorite kinkajou observations. And so that work with, a little bit of work with the olingo. Again, no one had hardly published anything about olingos, and so I was talking at the mammal meetings at the American Society of Mammalogists with Chris Helgen, and he said he had been looking at olingos in museum drawers and had found what he thought was a new species. It was smaller than olingos, it was redder, it had longer fur, and it lived really high in the Andes. And he was talking to me about it because I was one of the few people who had done anything with olingos. And my initial reaction was, we should go see if we can find it. And so we did, and we were able to find some of them up in the Andes, get some fresh tissue samples, and that's what we used along with the museum specimens to describe the new species, the olingito, which is in the same genus as olingo, so it's similar, but it's smaller. It's actually the smallest procyonid, and living high up in the cloud forest, again, where it's sort of smaller and has woollier fur that probably keeps it warmer and sort of more waterproof in the ever-present clouds.
So I sort of think of olingos as the sort of shy bookworm cousin that's like moves around fairly slowly, etc. And then the Kinkajous as a 4-year-old that's just drunk a bunch of Red Bull, right? I mean, they're just all over the place. Is that a fair, semi-fair description?
I think of them different. So the Olingos are smaller and they're much more agile, and so they will jump from tree to tree a lot more than Kinkajous. Kinkajous are heavier and they have the big prehensile tail. So a lot of time, what Kinkajous will do is they'll use their tail to hold on to the branch behind them, and then they'll lean way forward with their forearms, grab the next tree, pull that tree in until they grab a solid branch, and then let go with their tail and kind of spring forward. Whereas an Olingo doesn't have a prehensile tail, so they'll just kind of jump. And because they're lighter, they're about half the weight. Olingos are like a kilo or kilo and a half, and Kinkajous are more like 2 or 3. They seem to do better on that, and so they seem to be a little bit more agile. And often, like, I've spent a lot of time watching them at balsa trees. Balsa trees, if you ever can find a balsa tree in flower, which in Panama is at the very end of the wet season, beginning of the dry season, go there at night and you'll see all kinds of animals that come to drink the nectar from these balsa trap flowers. They're really big white flowers. And we spent a lot of time working with some film crews. We did a National Geographic magazine article on them as well. And so we spent a lot of time in these trees. And what we would see is the Olingo would get there, and when the Kinkajou would show up, the Kinkajou was kind of the boss and the dominant. And the Olingo would kind of run away or just feed at the edge and sort of jump out and jump back in to try to avoid the bigger Kinkajou.
And do they mostly eat the same things, or is there some sort of niche differentiation there?
Yeah, good question. They definitely both eat fruit and drink nectar of flowers. If you look at the teeth, it looks like the Olingo should eat some more insects. And I've looked at a lot of Kinkajou scats, and we didn't find any insects, like 0. Like there's some observations of them eating like ants or termites in Bolivia or something, but in my study in Panama, no insects. So the Olingos are probably eating some insects. We actually have some recent video of them of Olingos in Peru eating insects inside up in the canopy. So it looks like they're eating some more. We don't really know the details of like how important that is.
Oh, that's interesting. I'd no idea they were so much smaller. Whenever I see Olingos and Kinkajous, I just look at the tail. I'm just panicking trying to get a view of that to decide.
Yeah, it's tricky to tell them apart sometimes in poor light because they look about the same size.
Yeah.
And it's hard to see that, and they are like the length is pretty similar. Actually, the Kinkajous are just bulkier. One of the things I use to tell them apart is the Olingo has a more of a sort of bicolored, like a lighter belly and a darker back with kind of a lateral line that helps you see the difference. Whereas the Kinkajou is more continual. Like it's a little bit lighter on the belly, but it's more of a continual break. So often that's kind of the first thing I see when I'm trying to tell them apart. So there's that. There's the tail, as you say. The Kinkajou has a prehensile tail, so you can see them using it. Yeah. The Olingo tail looks about the same size just because it's bushy, but it's not muscular at all. And then the Olingo also has a pointier nose, if you can get a good enough look at its head.
Yeah. And are Olingos as vocal as Kinkajous?
Hard to say. To some extent, yeah, they both have their own noises, but it seems like Kinkajous might be a little bit more vocal.
Yeah.
Who knows what they're saying out there? I've had them yell at me quite a bit, or yell while I was underneath them. I couldn't tell if they were yelling at me or calling other animals in or scaring other animals away. Who knows?
Yeah, and I think I'm right. The Kinkajou's name comes from its call. Is that right?
I think it's an old native name. It did not. I call it a snortweedle. It's kind of a So that doesn't really sound like Kinkajou to me, but maybe to some people it
does. Right.
Yeah, fair enough.
Yeah. I can't imagine the logistics of trying to study Kinkajous at night crashing through the forests in Panama. You must have some good stories there. But
yeah, it was. You see a lot of cool mammals out there at night.
I bet you do. Yeah, really. That's the advantage of being out there. Yeah. So you you've obviously done a lot of research on carnivores, and in the US, you've also done a lot of work on coyotes, wolves, etc. And there was one paper that I read that you published, which showed that some of the weasels are suffering really large population declines across the US. Can you tell us a little bit more about that and what the causes might be?
Yeah, sure. So we were sort of concerned about weasels because, they're one of those animals that you just don't see very often, especially in the United States. I think in some areas you see them more often. And so we were just wondering, like, how are they doing? And there isn't really much systematic data that's collected on them. They're not easy to survey. And so we sort of we're just like, well, let's see what data there is out there. And one of the biggest data sets is fur trappers. And so if you look at fur trapping that's recorded by, the states and provinces, you see a huge decline in the harvest of weasels. Now that could be for a couple of different reasons, right? It could be because there's fewer people trapping weasels, which is definitely true. It could be because weasels are actually declining. And we did some models to try to take into account how many people were trapping, and we found that it was going down even more than you would think, just based on the number of people that are still running traps. Then we also looked at museum records and iNaturalist records of weasels. And the trick with those is they're opportunistic. So you don't know how much effort, you don't know how many people were out there looking, how many museums were out there collecting. So you have what you have, and we just looked at the patterns before and after 2000. And there are a number of areas that had a lot of museum records before 2000 that had very, very few or almost no museum records or iNaturalist records since 2000. And so that kind of highlighted some areas that it seems like weasels used to be a lot more common, but are a lot less common now. And so what we're trying to do now is get together a more systematic survey methodology and approach so that we can go in and evaluate and not just have these kind of ad hoc data sets, but actually get some better information on how the weasels are doing. Because, as to your questions of if they're declining, why, we have no idea. You could sort of tick off everything you could imagine. It could be pesticides, it could be habitat loss, it could be, there's a lot of owls out there these days, and owls often kill weasels. So maybe if you add lots of owls and then you've got all the white-tailed deer eating all the understory, there's less cover for them. So maybe it's just not as good habitat. Maybe it's climate change. We do see some of the declines are definitely in the hotter parts of the country. They seem to be holding on better in the mountains and some of the colder parts of the country. So it could be climate related, could be some combination of all those, but it's going to be hard to really figure out what the problem is and then try to address it without some more systematic data on them.
And is that all three species of weasels? Because in North America, you've got long-tail, short-tail, and the least. Did you see the declines pretty much across all of them?
Well, actually, no. The least and the short tail may be doing better up in the colder areas, and the long tail and some of the warmer parts of its range may be doing worse. Although it was also interesting when we looked at the least weasel, you look at the range map for least weasel, and it suggests they go all the way from sort of Ohio, even down into North Carolina, all the way up to Alaska. But if you actually plot the museum records and the iNaturalist observations on that, there's a huge gap between Alberta, Saskatchewan, and Alaska, or even like sort of northern, like North Dakota. So there's no record. It's like the range map says there are least weasels up there, but there are no records at all. And the least weasel, you probably know, lives in Europe and Asia as well. But it made us wonder if maybe the Alaskan weasel, the Alaskan least weasel, is related to the Eurasian one, and the North American one might be even a different species or certainly a different population, and something worth looking at a little bit more.
Yeah. And were you involved in the work that just split the short-tailed weasel has just been split from the US from the European one, I think, or just?
Well, they changed the genus. I think it was already split. But
oh, was it? Oh, I didn't realize. Genus. And there's also a new one on an island off of Alaska, I think. There's a.
Oh, could be. I missed that one.
Yeah, I know. There's all sorts of things have been happening with that recently, so
yeah,
it makes sense what you're saying. Yeah, interesting.
Yeah, it's funny because I hear very little about least weasels. if I just use sort of what comes across my desk in terms of photographs of the mustelids, so long-tail weasels, fairly often, and the short tails as well, but hardly ever do I see photographs of least weasels. They seem to be quite shy species.
Yeah, well, shy or super rare or declining or some combination of all three, right? And that's kind of our point, was like, hey, we need to figure out a better way. So what we're doing right now is testing, trying to figure out which stink do weasels like best. So we're putting out different combinations of scent lures and different combinations of meat bait and other kinds of bait to try to see, can we find a better way to survey them? And some folks are also trying some pretty neat drift fences. So often the way you catch reptiles is you put a fence, a little fence, like kind of knee height, and the animals walk along the fence and then they fall in a bucket. But instead of falling in a bucket, they put the bucket the other way around and put a camera trap looking down. So when the animal walks through the bucket, they get a picture close up from the camera trap. And they're getting weasels on those, which is really interesting, and small mammals, and snakes, and lizards, and all kinds of things. So it's kind of an interesting new approach that might be good for long-term monitoring.
Well, interesting. Yeah, very cool. And that leads us into the next question nicely, which is about your work with camera traps. I know you've become a huge fan of camera trapping. Can you talk about your work with Snapshot USA? What's it all about? And also, I know you've been doing some work using camera traps to try to better understand the relationship between fishes and porcupines, which are two very cool mammals.
Okay, yeah, two different questions.
Yeah.
So yeah, I love camera traps because they give you, a look at these mammals that are otherwise, many of them, very hard to see. And they also give you, it's sort of vouchered and verifiable, right? So if someone tells me they saw a least weasel, do I believe them? Maybe it was, it's hard to say for sure. And so I really like them for that. And then also you have this measure of effort. You know how long the camera trap was out for. And so that really adds the kinds of things you can do with those data compared to a random observation here and there. And so I've really been interested in using the data, archiving the data, and finding ways to get more cameras on the landscape, because what we need to track these animals, whether it's weasels or other species, and see how their populations are doing, how they're affected by climate change or other things. We need a lot of these dots on the map where we know what's there, how does it compare with this place, how does it compare with that place, how does it compare this year to last year. And so finding ways, we've done some work with citizen science, having volunteers run camera traps, and that works pretty well, but it's quite a bit of work, because it's not as easy as just recording an observation and uploading a photograph. It's more complicated. And so in Snapshot USA, what we've done is worked with a big network of scientists. And so we sort of recruited mammalogists all across the country and say, hey, who's got camera traps already and wants to run them all together at a national survey in September and October? So everyone runs cameras, nearby their house, or their, not in their house, but like, in a local forest preserve or even an urban area. So we get all kinds of different habitats. We all run them in following the basic same protocol. And then we upload the data. We use Wildlife Insights to manage the data. We all upload the data to the same place. We code the data ourselves. So often this is done with classes. University classes often are involved, but not always. And then we do some quality control, and then we publish the data. And we make the data publicly available to anyone who wants to use it. Everyone who helped with the study is a co-author on the paper. And then the next year we do it again. And so we started in 2019 and had a great response. We were able to get cameras in all 50 states that year, and we've been able to do it every year. Some studies have added, some studies have fallen off. But this is we're looking at it as a model for sustainable monitoring of mammal populations. We get some turkeys and some other stuff, but it's mostly the mammals. As a way to get regular monitoring every year. And we've now started having some interest from other places. We've done Snapshot Europe twice now, and had talking to some colleagues who are interested in maybe starting it up in some other countries as well.
Really cool. Yeah, I'm struck by how the field has advanced so much over the last few years. And when I think about camera trapping it feels like just a few years ago, it was these blurry images of things. And now, it's a category of its own in the Wildlife Photographer of the Year competition. These extraordinary photos of animals, which take just as much skill. There's a bit of an argument going on among photographers about whether camera trapping pictures should be included. And yet some of those pictures have got maybe more skill than in the setup than sitting there with your finger on the button. And then there's all the scientific benefits, and it's fantastic.
Yeah, without a doubt, those professional camera trap setups are not simple, and they take a lot of mammal knowledge, because when they set up those the fancy ones, they need to know they need to focus the lens where they think the mammal's going to go, and so they need to have they need to understand how the mammal sees the world, how they're moving, where they think they're going to be going, and get the light. And then they have the flashes on the side. It's really it takes a lot of knowledge and skill, and then to get it artistic, a bit of artistic flair as well.
Yeah, for sure.
Yeah. But it was, it was only just 15 years ago that we were using the old deer cameras, where you put old film, 36 shots in it, etc. And then one baboon troop in East Africa walk past your camera trap. That's it. You're done. You're done until you change it out next month. So now they take 10,000 shots each or more. So it's
well, and then we're also working on the AI to help filter through them. We've got it working pretty well now, where it's in Wildlife Insights, it'll tell you what it thinks it is, and then you can confirm it or change it. And it's pretty good for a lot of things. The first time someone uploads camera traps from pictures from New Guinea, of course, it's not going to know anything. But once we get those images in there, then it'll be trained up, and it'll be able to work on those species as well.
Oh, cool! Fantastic.
Oh, so porcupines and fishers.
Yeah, the second part of my question. Yeah.
So fishers are one of my favorite species, and I had the fortune to study them some when I was living in Albany. And I'm always amazed by the fisher-porcupine interaction, right? Because fishers are one of the only mammals that regularly kills porcupines. I think cougars do it pretty often, but otherwise, that's about it. In North America, the fisher is the enemy of the porcupine. And as far as I know, no one's ever seen it in the wild, and no one's ever filmed it in the wild. Maybe some of your listeners will correct me. I've certainly never seen anything on YouTube. There was a biologist in the 70s that kind of set it up in a scene and saw what happened, and other people have found carcasses. We found a carcass that was almost certainly killed by a fisher. So what we think they need to do is that the fishers have to catch the porcupines on the ground. So especially in the winter, the porcupines will sleep in a hole or in a rock crevice or something, and then they'll walk through the snow and then go up a tree to eat. And so when they're in their hole, they're safe, and we're in the tree, they're pretty safe. But we think the fisher needs to get them on the ground and then sort of run around and bite them on the head. And so I wanted to try to film this, and I went to a couple of different places outside of Albany, and you can see the porcupine trails really well in the snow. And so I would just set camera traps up all along the trails to try to get it. And we got tons of footage of porcupines, which are now also one of my favorite animals. I had never seen them so up close and so personal, and learned so much about them as this experience, and also lots of fishers. We never got them actually fighting with each other. So I put up a YouTube video on. I started a YouTube channel called Wild Animals, and so you can watch the what I learned about fishers and porcupines. And we did get them a couple times. I had a camera on a porcupine in a den, and the fisher would come up and kind of sniff it and see just a quills and run away. So it's something I'm going to have to try again. I was hoping to do it this winter, but we didn't get the right snow conditions, unfortunately.
I was obsessed with fishers for many years, and you won't remember, but I actually emailed you and you gave me some advice several years ago about looking around Albany for them.
Oh yeah, I remember that.
You might remember.
Oh, yeah.
So
did you find any?
I eventually saw one in Algonquin. If they get any roadkill in the winter, they put it in front of the visitor center and stick a camera up on it, and inevitably fishers will start feeding on it. So I went up there, but I had a long and sorry story of misadventures on my quest, which included speeding tickets, losing my rental car keys in the snow.
Oh no!
Anyway, that's a whole story. But I was always looking for them in trees, and I'd always assumed they were arboreal hunters. But you're saying they feed on the ground.
Yeah, I mean they're decent. They're decent climbers.
Yeah.
And when we track them in Albany with GPS, and one of my favorite things in the world is to jump on a Fisher track in the snow and follow them and see what they're up to. And we didn't really see them jumping from tree to tree very often. They would go up. What they would do, the only hunting I saw them do in the trees is in the winter the gray squirrels are often in their leaf nests, their drays, and the Fishers would go up there. We called it a Fisher bed and breakfast because they would kind of kill the squirrel and then lay up there and sleep. So we would often find them sleeping on top of one of those leaf nests. So, and could they attack a porcupine in the tree? For sure. But I think that's going to be tough because the porcupine can just turn around and then you just got the back full of quills. Yeah. There's also an amazing video on YouTube of a Fisher chasing a squirrel up and down a tree.
Oh, really?
I'm sure it'll show up. And it's amazing to me the squirrel doesn't just run to the top of the tree to run away. He stays on the trunk and just does the kind of spin around to the other side game. And eventually the squirrel loses.
Oh, wow. I'll check that out. Cool.
Yeah, that's great. So, and the European sable, is that effectively the same species as a Fisher? I mean, is it exactly the same?
No, Fisher's quite a bit larger.
Fisher's bigger, is it? Yeah. Okay. Yeah. Right.
Yeah. So, the female Fishers are like 2 kilos or so, but the males, we had one that was almost 6 kilos. That's a pretty big animal.
Right. Yeah. I wanted to ask you a little bit about your work on the coyote hybridization that you did, certainly in the New York area. Can you tell us a bit about that?
Yeah, sure. So, coyotes were originally only living in the Western United States, in basically open country of the Western United States and Mexico. They didn't get down into the tropical rainforest. They didn't get up into the Pacific Northwest rainforest. They didn't get up into the eastern deciduous forest or the great northern forest. And then they did, right? And then they colonized starting about 1920. They moved in all directions. And we don't know exactly why. Part of it's probably because we killed the wolves in a lot of those areas, and in some of the areas, the mountain lion. So the lack of a competitor and predator might have helped them. There's certainly a lot of forest fragmentation that opened up the type of habitat that they prefer. So that was almost certainly part of it. And so the coyote moved in, first moved into upstate New York in kind of the 1950s. Here in Raleigh, here in North Carolina, it didn't get here until really the 80s or 90s in some areas. And along the way, they started looking different. And so, when they first got into New York, people called them coy dogs because, implying that they had hybridized with dogs. And other places, they call them coy wolf. And so, and they definitely look different. And so we did one of the first systematic surveys, and there's been a lot of work since then as well. And what we have learned is that the coyote hybridized with wolves first, probably in the Great Lakes. And if you think about when two species hybridize, they don't really want to, right? They would rather breed with their own type. And when we think it happens is when one is really rare and can't find another animal to breed with. And so if you think about 100 years ago in Minnesota and the Great Lakes, there were a few wolves barely hanging on. And that's when it looks like they hybridized with coyotes. And so then you have an animal that's 50-50, right? That first animal is going to be an F1 hybrid. Its mom was probably a wolf and its dad was a coyote. But then that animal is fertile. So all the canis can breed. Dogs, wolves, and coyotes can all breed with each other and have fertile offspring. And then those puppies, those would have grown up and they would have then bred with another animal, either a coyote or a wolf. And so now that offspring is going to be 25-75. And then that's going to cross again and going to become, 12.5 to 80%. And so what happened is those wolf genes worked their way into the coyote population. And those coyotes that had the wolf genes in them, they moved very quickly east. And they were significantly larger, and their skulls were larger and especially extra wide. And by having an extra wide skull, you have more room for muscle attachment and a much stronger bite. which might have helped them hunt deer. And so those animals moved into the east 5 times faster than the animals that were down in Pennsylvania, for example, that didn't have wolf genes, that were also moving east, but they were moving east a lot slower. So the ones that went up through Ontario and then down into New York and New England moved much faster, and they had more wolf genes. And what we found, somewhere along the line, they also hybridized with dogs, and probably multiple times. And we think it's the same phenomenon where if you imagine the first coyote crosses into upstate New York, there's no coyotes behind it, or there's no other coyotes there, and the other coyotes are pretty far behind. So if she wants to breed, they're very seasonal breeders. She's got to take the next best thing she can get, which would have been domestic dogs running around. And so that's probably when the dog hybridization occurred. Nowadays, in most parts of the United States, there's coyotes everywhere. And so they don't hybridize with dogs anymore, but there's still some of those genes that got mixed in. And so we did some pretty detailed genetics on a bunch of animals from the Northeast United States, New York, and other parts of New England. And we found on average, they were about something like 8% dog, 8% wolf, and the rest coyote. So they're still mostly coyotes, but they've got some dog genes mixed in. They've got some wolf genes mixed in. You can look at the details of the genetics and see if those genes are being selected for. So do they show up more or less than you would expect on average? If they were helping the animal survive, you would see them more than on average. And if they were hurting the animal survival, you would see them less on average. And we found the wolf genes were positively helping the coyote survival. And many of those, you don't know exactly what they do, but from the dog genome, you have some idea of what kinds of things those genes probably do. And many of them were related to growth and body size. So we think that we have pretty good evidence that the wolf genes have helped the coyotes get a little bit bigger. They're like 10 pounds heavier than Western coyotes. So it's not like they're wolf size yet. They're still sort of, 35, 45 pounds. Very rarely you'll get a 50-pound animal. I haven't seen much in the coyotes that are actually heavier than 50. So they're a little bit bigger. Wolves are going to be much larger. What the dog genes are doing, we don't really know. We haven't sort of looked into what those are doing. It does seem to me that you see more funny colored coyotes in the East, red colors, black colors. Seen some animals with some really fluffy hair that just kind of looks ridiculous, doesn't look like a coyote at all from the hair. And so my guess is that those are dog genes that are sort of showing up that are mixed. You're going to get different, depending on the combinations, you could get all kinds of different things showing up. And so it seems like the dog genes and the wolf genes are really well spread across the East, that there's no more hybridization with dogs except in Long Island, which is really interesting right now because Long Island is the only, until recently, so coyotes just got there. And it's really hard to get to Long Island, right? There's all this development, there's all these roads and bridges and tons of traffic. And so there haven't been coyotes there for a long time. And they recently established, and there was definitely a litter that was a first generation dog hybrid litter. They have camera trap pictures of it. And so if you think about the scenarios that I was talking about earlier, there were coyotes that would show up in upstate New York and they would hybridize with dogs. And then all these other coyotes would show up. And so they would back cross into the coyotes. But in Long Island, you don't have that because it's still going to be hard for the coyotes to get there, right? So I think I'm really interested to watch it to see if Long Island develops like a proper coy dog. Proper, I mean, with substantial amounts of dog genes and coyote genes, as compared to everywhere else where eventually the coyote genes would kind of swamp it out. So this is sort of happening right now. And so I'm definitely keeping an eye on the coyote photographs that come out of Long Island nowadays.
Wow, that's just fascinating. It's
Yeah.
and the Western coyotes, do they have any evidence of hybridization?
No, not that anyone has seen. There's no genetic evidence of it. There's lots of coyote studies of Western coyotes, and they don't find any hybridization evidence with wolves or dogs. The wolf population's higher, so you might not expect that as much. Panama, though, is another case. We have been running some cameras down in Panama, and there are some funny looking coyotes there. And we actually have a photograph of one hybridizing with a dog. So we don't have any genetic results from that project yet. But I wouldn't be surprised if in Latin America they're hybridizing with dogs. And we have the coyotes now at the very doorstep of South America. We have them at the edge of the forest at the Darien Gap. And so I never bet against coyotes, and I think it's only a matter of time till they show up in South America.
Wow. So it seems to be a feature that's linked to range expansion, the hybridization.
Yeah, probably linked to range expansion and linked to being rare, and so not having another of your kind to breed with, and so sort of taking advantage of whatever else is available there.
Right.
The other fun thing about Panama is the crab-eating fox from South America has already moved north and crossed the Darien Gap and moved at least to Panama City. So we have two canids, the crab-eating fox and the coyote, that have never in evolutionary history ever bumped into each other, that are presently overlapping in Panama right now.
All right.
Wow.
Interesting.
That's
and was it you, Roland, that came up with the term Canis supus for this species?
I have used it a lot.
Yeah.
I think somebody else made it up. I can't remember who it was. It might have been someone from Defenders of Wildlife. It was quite a long time ago. So it's a great term. I think it defines what's going on in North American canids, coyotes and wolves and red wolves in a nice, simple way that people kind of can make sense of.
Exactly. Yeah.
So you've spent a lot of time wandering around the forests of South America at night, and obviously you work in many, many different countries. You must have seen some quite amazing animal encounters. Can you tell us a few of those?
Oh, sure. Yeah. So, we mentioned spending a lot of time out at night with kinkajous, sometimes with a field assistant, often by myself. And so, you see a lot of cool animals out there. And some of my best observations were during a rain or after a rain, because that's where everything's either, if it's raining, the animals can't hear you, or if it's just everything's wet, the leaves don't crunch as much. And so you can walk a lot quieter. And one of my favorites, I was coming back towards the truck at probably a little after midnight, and it had been rainy, and I saw these two eye shines, and they're staring straight at my headlight, and which kind of blinds them, right? If they stare straight at your light, they can't really see. And so I was able to slowly walk up because it was so quiet, and I got about 2 or 3 feet away from a pair of jaguarundis that were just sitting there and just staring straight at me. And I actually got a little bit nervous because I was afraid, what if they freak out and jump on my face or something like that? I was that close. So I had my telemetry antenna out there between me and them, just in case. So that was really cool, because just such a cool cat. And I've seen them other times, but just glimpses as they're running away. And so it was just really special to get such a close-up look at the two of them and have them sort of staring straight at me, trying to figure out what the heck I was.
Oh, that's great.
Magic. It does seem that just about every sighting is a fleeting glimpse of one running across the trail. So that was very, very lucky to have that experience. And they're such a strange looking cat as well.
It is such a strange looking head. Not like any of the other felis, right? Yeah, I think they're closest related to pumas, actually.
Yeah, but they look more like the Fusa from Madagascar, I was thinking. Not that I've seen the Fusa, that's a sore point. But yeah. So you've written a field guide, which I have. I also have your fantastic app, which I love, particularly because you can select what area you're in of the country to narrow down the choices. But you wrote the field guide with Don Wilson. What was your motivation for doing that? And how long did it take?
Yeah, well, so I was in grad school, and I was really had gotten into bird watching. I had some buddies in grad school that were bird watchers, and whenever I travel to a new part of the world, I was always excited to get the bird guide and see what birds are there and go find out, try to find them and just learn about the birds. And it was amazing to me, at the time, the North American Mammal Field Guide was an old Peterson guide by Bert from like basically the 60s. I think it had been revised once or something like that. And so I was just thinking like someone should do another field guide. And so I just wrote to Don. I didn't know Don Wilson very well, I think at all at the time. Maybe I met him at a mammal meeting once. I had a guy I was in grad school with knew him and encouraged me because, you know, Don was a super big name at the Smithsonian. And he said, Don's very approachable. He's very friendly. You should just write him. And so I wrote him and he was keen. And so we, you know, I relied on him for especially sort of navigating the publishing world and stuff like that. And we of course first went with Peterson, but as you may know, Fiona Reed wrote a book for them, so they had signed with her recently. So we ended up doing it with Princeton University Press, which is a fantastic place to write to do field guides. And so that was really a big library project for me, and some museum work and a little bit of field work. But you know, the mammals of North America are pretty well documented. And so a lot of it was about, you know, learning about the animals, especially learning, you know, how do you tell them apart? What's the key characteristic to tell them apart? And, you know, sometimes it's a skull character. So we tried to show some of those. We did have one fantastic field trip with a lot of the artists that did some of the artwork down in Portal, Arizona at the Southwest Research Station. And we hung out there for a week and trapped a bunch of animals and helped them put them in, you know, aquariums, or they had a bunch of cages and sort of holding pens that we could catch animals and just let the artists sit there and paint them and work with them. So that was definitely a highlight in the sort of prep work for that.
And how much would you rely on museums? Because, I mean, at the time when you were writing it, if I'm correct, iNaturalist wasn't around.
Correct.
So you would have to rely on scientific papers, museums, etc. Was that your main source of information for your book?
Well, yeah. So the artist, for sure, used the specimens, you know, because they're painting them. And so they had to get that just right. For me, it was more a library project, you know, because somebody else has already figured out how do you tell these species apart. So I didn't need to go to the museum and lay all the specimens out and figure that out for myself. I had to read what other people had done, and then I would highlight to the artist, you know, okay, on these kangaroo rats, we need to really show the tails and emphasize the differences between the tails because that's the key difference to tell them apart. And on this species, you know, it's the underside of the hind foot or whatever. And so, you know, sort of my job was to figure out what those differences were and highlight them to the artist and figure out, you know, which species do we want to have together on the same plate so they're easy to compare.
Right. And in terms of pulling together the range maps, was that based on a lot of known information, or how did you?
Yeah, fortunately, when we were doing this was right around when kind of the global mammal community was starting to pull together range maps in a standardized way. Mm-hmm. And so that was something I was able to tap into some of those IUCN maps rather than try to do that myself, because back then that would have been really hard. You know, it really was a whole community of mammalogists who made those maps. It was not something that one person could do, you know, at the time. Nowadays, the data is at your fingertips, and so it would be a little bit easier.
Yeah. So, Roland, you also run a podcast, also about wildlife. Can you inform our listeners a little bit about the topic of your podcast and what it is that you typically ask your guests about?
Yeah, sure. So the podcast is called Wild Animals, and it's inspired by my own work radio tracking animals like the kinkajous or the fishers that we talked about before, and how you get to know them individually and you get to kind of learn about the drama of their lives, how they're interacting with other species, what they're going through. And so I wanted to find a way to tell those stories, because it's really a privilege, I think, for a scientist to, you know, get to know individual animals like that. And it's pretty common in animal tracking, where you're putting a tracking device on, maybe a GPS or a VHF, and you're following, you know, the individuals around. Often you give them names, so you kind of get to know them. And, you know, whenever the grad students are getting together, they're always like, oh, what did you find today? You know, what happened? And so I wanted to find a way to share those stories. And I thought the podcast sort of platform was a good way for that. And so I interview biologists who have interesting stories about animals that they're studying, and we kind of review the what is the species, what is the what was the research project that you're doing. So you get to learn about the research, you get to learn about the field work, about the trials and tribulations of doing the work. But then really, it's sort of like, well, who was the animal in question, and how did you first meet them, and what happened next, and what happened next, and what happened next, and to sort of tell the story of that animal, and then ending, and how did it end, and maybe they just got a GPS download from them yesterday and they're still doing great, or maybe the animal died in some way, or whatever. So there's sort of different endings to the stories, and so we're in season 3 right now, which is exciting. We had Waffles, the hyena who climbed the social ladder, first up. And coming up, we've got a number of mammals, including Cheetah 784, the orphan who overcame, Kilkenny, the red wolf who hatched the hypothesis, and I think one of my favorites of the season, it'll be the last one, it's WDF 128, the African wild dog who was a phoenix. It's really an incredible story about literally an animal who was the phoenix for this population.
Wow. Oh wow! You have the best titles. They're fantastic.
You do,
yeah.
Yes.
I want to listen to all of these.
Hours thinking about
much better than our season 2, episode 6.
I think we need to work on that, John.
That's part of the trick, because some of the animal stories are not nearly as interesting, right? So it's always kind of talking to the biologists and being like, what's the best story, what's the best animal, and usually they tell stories of more than one. It's kind of one animal's the star of the show, but they end up talking about some other characters as well.
Right.
Sounds excellent. Okay, we'll definitely put a link to this in the notes and full details on how people can find this. I'm going to listen.
Yeah.
Now, Roland, thank you so much. That was an absolute joy to listen to your stories and to hear you explain what a very complex idea is so simply. You're a great communicator. I will be tuning into your podcast. I'm sure many other people who listen to this one will be. Sounds wonderful. Thank you so much for sparing the time to join us, and good luck with all you're doing.
Thanks so much for having me on here. It's really fun.
Yeah, it's been hugely entertaining listening to your stories, and I will definitely be following your YouTube channel when you actually get to find a picture of a fisher eating a porcupine. That's what I really want to see.
Thanks. Well, that might be next time, but we've got a couple great episodes. One, we put out a bunch of baby fawn decoys to see what would attack them. That's coming up soon, and it's pretty awesome. We fooled a lot of predators.
Probably a lot of hunters too. Yeah.
You've been listening to Mammal Watching with Charles Foley and John Hall. You can find other episodes at mammalwatching.com/podcast.